in which we will study about metabolic fates of pyruvate, the master switch of metabolism. If you're asked to discuss the metabolic fates of pyruvate, describe its transport from cytosol to mitochondria, elaborate the reaction catalyzed by the pyruvate dehydrogenase complex, along with its regulation and significance, or enlist inherited and acquired causes of lactic acidosis and give the biochemical explanation in each case, then here's exactly what you need to know. Pyruvate, the end product of glycolysis, is a crucial metabolic intersection. Depending on oxygen availability and cellular demands, it follows 5 major fates. First, it can be converted to acetyl-CoA by the pyruvate dehydrogenase complex in the presence of oxygen. This enters the TCA cycle or is used for fatty acid synthesis. Second, in anaerobic conditions such as in red blood cells or actively contracting muscles, pyruvate is reduced to lactate by lactate dehydrogenase to regenerate NAD+ for glycolysis. Third, pyruvate can be converted into oxaloacetate by pyruvate carboxylase, playing a key role in gluconeogenesis and replenishing TCA cycle intermediates. Fourth, it can be transaminated to alanine via ALT, especially during fasting, as part of the alanine cycle transporting nitrogen to the liver. And fifth, in organisms like yeast, pyruvate can be converted to ethanol through fermentation, a pathway that doesn't occur in humans. Now for another key question. How does pyruvate enter mitochondria? Pyruvate is produced in the cytosol and transported into the mitochondrial matrix by a pyruvate H+ symporter. This step is critical for its aerobic metabolism via the PDH complex. Speaking of the PDH complex, let's go over its reaction, regulation, and significance. The complex catalyzes an irreversible reaction. Pyruvate combines with CoA and NAD+ to form acetyl-CoA, NADH, and CO2. This takes place in the mitochondrial matrix and is facilitated by a multi-enzyme system. The reaction requires 5 essential cofactors: thiamine, vitamin B1, lipoic acid, CoA from vitamin B5, FAD, vitamin B2, and NAD+, vitamin B3. Use the mnemonic TLC for Nancy to remember them. The PDH complex is tightly regulated. It is inhibited by high levels of ATP, NADH, and acetyl-CoA, and activated by ADP, NAD+, CoA, and calcium. It's also subject to covalent regulation. PDH kinase inactivates it via phosphorylation, while PDH phosphatase reactivates it, especially in muscle cells during contraction when calcium levels rise. The importance of this step cannot be overstated. It links glycolysis to the TCA cycle and commits pyruvate to aerobic respiration. And since it's irreversible, pyruvate cannot be converted back into glucose once this step occurs. Now let's look at lactic acidosis, a common clinical and exam topic. Lactic acidosis occurs when pyruvate cannot enter aerobic metabolism and is instead diverted to lactate, causing elevated lactate levels and decreased blood pH. There are both inherited and acquired causes. Inherited causes include PDH complex deficiency, which is X-linked. This leads to pyruvate buildup and conversion to lactate, causing high lactate, high alanine, and normal glucose levels, often presenting with neurological symptoms due to impaired energy production. Another inherited cause is mitochondrial disorders that impair oxidative phosphorylation, causing NADH accumulation, which inhibits PDH and shifts metabolism toward lactate. Acquired causes include thiamine deficiency, often seen in alcoholics or malnourished individuals. Thiamine is required for PDH function, so its deficiency mimics PDH inactivity. Hypoxia or ischemia reduces oxygen supply, impairing electron transport and increasing NADH, which again inhibits PDH and favors lactate production. In sepsis or shock, tissue perfusion drops, anaerobic glycolysis increases, and lactate accumulates. Lastly, toxins like arsenic, which inhibits lipoic acid, and poisons like cyanide or carbon monoxide, which block the electron transport chain, also lead to NADH buildup and lactate accumulation. So here's your final recap. Pyruvate has 5 main fates: acetyl-CoA, lactate, oxaloacetate, alanine, and ethanol. It is transported into mitochondria via a proton symporter. The PDH complex converts pyruvate into acetyl-CoA using 5 vitamin-derived cofactors and is regulated both allosterically and by phosphorylation. Lactic acidosis results from impaired aerobic metabolism of pyruvate, whether inherited or acquired, and always involves a shift toward anaerobic lactate production. To lock this in for your SEQ or Viva, quiz yourself. Name the 5 fates of pyruvate. Which vitamin deficiency mimics PDH deficiency? Which toxin blocks lipoic acid? What activates PDH in muscle? And which condition causes high lactate with normal glucose? Say it aloud, write it down, or ask a friend. That's how you turn facts into recall power on exam day. Now we will talk about the TCA cycle, also known as the Krebs cycle or citric acid cycle, is the final common pathway for the oxidation of carbohydrates, fats, and proteins. It takes place in the mitochondrial matrix and begins only after pyruvic acid is converted to acetyl-CoA by the pyruvate dehydrogenase complex, a frequently tested MCQ. Although oxygen is not used directly in the reactions of the cycle, it is still essential for the regeneration of NAD+ and FAD through the electron transport chain. The cycle starts when acetyl-CoA combines with oxaloacetate to form citrate. Through a series of enzyme-catalyzed steps, citrate is gradually converted back into oxaloacetate, releasing high-energy molecules along the way. The main intermediates include citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and finally oxaloacetate. It's important to remember that pyruvate is not an intermediate of the TCA cycle, a commonly asked MCQ. 3 key steps in the TCA cycle produce ATP or its equivalents. First, the conversion of isocitrate to alpha-ketoglutarate by isocitrate dehydrogenase generates NADH. Second, alpha-ketoglutarate is converted to succinyl-CoA, producing another NADH. And third, succinyl-CoA is converted to succinate, yielding GTP, which is equivalent to 1 ATP through substrate-level phosphorylation. Later reactions generate FADH2 and more NADH. In total, 1 round of the TCA cycle from a single acetyl-CoA molecule produces 3 NADH equal to 7.5 ATP, 1 FADH2, 1.5 ATP, and 1 GTP, 1 ATP, giving a total energy yield of approximately 10 to 12 ATP, another high-yield MCQ. The cycle is tightly regulated based on the cell's energy needs. It is activated by ADP and calcium ions and inhibited by ATP, NADH, and certain cycle intermediates. Specific enzyme checkpoints include citrate synthase, which is inhibited by ATP, NADH, citrate, and succinyl-CoA. Isocitrate dehydrogenase is inhibited by ATP and NADH. Alpha-ketoglutarate dehydrogenase is inhibited by NADH, succinyl-CoA, and arsenite. And succinate dehydrogenase, another high-yield MCQ, is competitively inhibited by malonate. These inhibitors ensure that the cycle slows down when energy levels are high, preventing unnecessary oxidation of fuel. Now let's talk about the significance of the TCA cycle, a favorite question in SEQs and Viva. This cycle is the hub of metabolism. It not only produces ATP and reducing equivalents, but also provides intermediates for biosynthetic processes. For example, alpha-ketoglutarate is a precursor for amino acids, succinyl-CoA is used in hemosynthesis, and citrate plays a role in fatty acid synthesis. The regeneration of oxaloacetate is crucial to keep the cycle running, another commonly asked concept in exams. Another frequent SEQ asks, what is the role of B-complex vitamins in the TCA cycle? These vitamins function as vital coenzymes. Thiamine, or vitamin B1, is essential for alpha-ketoglutarate dehydrogenase. Riboflavin, or B2, forms part of FAD. Niacin, or B3, is used to form NAD+. Pantothenic acid, or B5, is a component of coenzyme A. Biotin, or B7, indirectly supports the replenishment of oxaloacetate by activating pyruvate carboxylase. Without these vitamins, the cycle slows down or halts entirely, leading to serious energy deficits and possible metabolic disorders. To test your understanding, ask yourself, which steps of the TCA cycle produce NADH, FADH2, or GTP? What happens when vitamin B1 is deficient? Which enzyme is inhibited by malonate? Why is oxygen needed even though it's not directly used? And how many ATPs are produced per acetyl-CoA? In summary, the TCA cycle is a central aerobic metabolic pathway that connects catabolism and anabolism. It's regulated by energy signals, requires essential vitamins, and is influenced by specific inhibitors. To truly master this topic, focus on understanding the why behind each reaction. Let's dive into one of the most high-yield exam favorite topics in biochemistry, gluconeogenesis, a major topic of UHS MCQs, SEQs, and VIVAs. Gluconeogenesis is the metabolic process where glucose is synthesized from non-carbohydrate sources, especially during fasting, starvation, prolonged exercise, or diabetes. It mainly occurs in the liver, and to a lesser extent in the renal cortex. This pathway is suppressed after feeding due to insulin and is stimulated during fasting by glucagon and cortisol. According to UHS MCQs, gluconeogenesis is simply the synthesis of glucose from non-carbohydrate precursors. So, what are these precursors? Common substrates include lactate from anaerobic glycolysis, alanine from muscle protein breakdown, glycerol from triglycerides in adipose tissue, propionyl-CoA from odd-chain fatty acid oxidation, and TCA cycle intermediates like malate and oxaloacetate. A common UHS MCQ asks, which of these is not a gluconeogenic substrate? The answer is acetyl-CoA. Now let's look at how each precursor feeds into this pathway. Lactate is converted to pyruvate via lactate dehydrogenase and enters gluconeogenesis through the Cori cycle, where lactate from muscles is transported to the liver and converted back to glucose. Alanine is converted to pyruvate via alanine aminotransferase in the glucose-alanine cycle, which not only supplies glucose but also helps detoxify ammonia. Glycerol is converted to glycerol 3-phosphate, then to DHAP, and eventually enters the gluconeogenic pathway. Since glycolysis has 3 irreversible steps, gluconeogenesis bypasses them using alternate enzymes. Pyruvate is converted to oxaloacetate by pyruvate carboxylase, a biotin, vitamin B7-dependent enzyme and the rate-limiting step of gluconeogenesis. Oxaloacetate is then converted to phosphenylpyruvate by PEP carboxykinase. Later, fructose 1,6 bisphosphate is converted to fructose 6-phosphate via fructose 1,6 bisphosphatase, a step inhibited by fructose 2,6 bisphosphate. Finally, glucose 6-phosphatase converts glucose 6-phosphate to free glucose, an enzyme only found in the liver and kidney, explaining why muscles cannot perform gluconeogenesis. Regulation-wise, gluconeogenesis is enhanced by fasting, high ATP, glucagon, cortisol, and acetyl-CoA. It is inhibited by insulin, AMP, and fructose 2,6 bisphosphate. According to UHS MCQs, which hormone inhibits gluconeogenesis? The answer is insulin. And the molecule that allosterically inhibits it? Fructose 2,6 bisphosphate. Let's revisit the 2 important cycles. The Cori cycle transports lactate from muscle to liver for conversion to glucose, preventing lactic acidosis and supplying energy. The glucose-alanine cycle transfers alanine from muscle to liver, converting it to glucose and removing ammonia via the urea cycle. These cycles frequently show up in both SEQs and MCQs. Clinically and exam-wise, remember these key points. The 3 enzymes that bypass glycolysis are pyruvate carboxylase, fructose 1,6 bisphosphatase, and glucose 6-phosphatase. Biotin deficiency halts gluconeogenesis due to its role in pyruvate carboxylase activity. During fasting, energy is derived from fat breakdown, which provides ATP and glycerol, and muscle breakdown, which gives alanine. Lactate from anaerobic metabolism also contributes. The tissues capable of gluconeogenesis are primarily the liver and kidney, not muscles. To close this up, gluconeogenesis is a hormone-regulated, energy-dependent, life-saving process. It supplies glucose when carbohydrates are unavailable, uses substrates like lactate, alanine, and glycerol, operates under tight hormonal control, integrates with Cori and glucose-alanine cycles, and depends on coenzymes like biotin and key enzymes like PEP and pyruvate carboxylase. Understand the logic, the steps, the regulators, and you'll ace every MCQ, SEQ, and Viva. In the world of carbohydrate metabolism, glycogen metabolism is a frequently tested and clinically important concept. It includes glycogenesis, the synthesis of glycogen, and glycogenolysis, the breakdown of glycogen. These reactions are especially vital during fasting, exercise, and times of energy demand. Glycogenesis, which primarily occurs in the liver and skeletal muscle, begins with glucose being phosphorylated to glucose 6-phosphate by either hexokinase or glucokinase. This is then converted to glucose 1-phosphate by the enzyme phosphoglucomutase. Glucose 1-phosphate reacts with UTP to form UDP glucose, the activated glucose donor. Glycogen synthase adds UDP glucose to a growing glycogen chain, while the branching enzyme forms alpha-1-6 glycosidic bonds, increasing the solubility and accessibility of glycogen. According to UHS past paper questions, one common SEQ asks, how is glycogen synthesized in the liver? The regulation of glycogenesis is both hormonal and allosteric. Insulin activates glycogen synthase through dephosphorylation, encouraging glycogen storage after meals. In contrast, glucagon and epinephrine inhibit glycogen synthesis. Another important UHS SEQ asks, how is glycogen synthesis regulated? A related MCQ asks, glycogen synthase is activated by? And the correct answer is dephosphorylation, which is stimulated by insulin. Allosteric regulation ensures that glycogen synthesis and degradation do not happen at the same time. Phosphorylation activates glycogen phosphorylase for breakdown and inactivates glycogen synthase for synthesis. Insulin promotes dephosphorylation, while glucagon and epinephrine promote phosphorylation. Allosteric regulation also plays a key role. ATP, glucose 6-phosphate, and citrate promote glycogenesis. Meanwhile, AMP and calcium stimulate glycogenolysis, particularly during muscle contraction. So, be prepared to answer, how is glycogen synthesis and degradation allosterically regulated in the liver and muscle? This is another commonly asked UHS SEQ. What is reciprocal regulation of glycogen metabolism? Now, shifting to glycogenolysis, this is the process of breaking down glycogen when the body needs glucose. Glycogen phosphorylase cleaves glucose residues from the glycogen chain, releasing glucose 1-phosphate. Then, the debranching enzyme remodels the structure by transferring glucose chains and removing alpha-1-6 branches. The released glucose 1-phosphate is converted to glucose 6-phosphate by phosphoglucomutase. In the liver, glucose 6-phosphate is dephosphorylated by glucose 6-phosphatase to produce free glucose that enters the bloodstream. UHS SEQ exams often ask, outline the steps of glycogen breakdown. Another important question is, why is only a little amount of free glucose formed from glycogen degradation in muscles? The answer is, muscle lacks glucose 6-phosphatase, so glucose 6-phosphate enters glycolysis directly instead of being released into the blood. A past MCQ reinforces this point. Muscle glycogen cannot contribute directly to blood glucose because of, and the answer is, absence of glucose 6-phosphatase. Let's now talk about glycogen storage diseases, a high-yield exam topic. These are inherited enzyme deficiencies that impair glycogen metabolism. UHS SEQs often ask you to enlist glycogen storage diseases or explain their biochemical defects. Here are the main types. Type Y, von Gierke's disease. Caused by a deficiency in glucose 6-phosphatase, it leads to severe hypoglycemia, hepatomegaly, and lactic acidosis. A past MCQ asks, von Gierke's disease is due to the absence of which enzyme? The correct answer is glucose 6-phosphatase. Type 2, Pompe's disease, caused by lysosomal acid maltase deficiency. Features include cardiomegaly, hypotonia, and often fatal outcomes in infancy. Type 3, Cory's disease, due to debranching enzyme deficiency. It causes glycogen with short outer branches and mild hypoglycemia. A clinical case SEQ describes a 4-month-old infant with hypoglycemia and hepatomegaly. A liver biopsy shows glycogen with short branches, pointing to Cory's disease. Type AFU, Anderson's disease, caused by a deficiency in the branching enzyme. This leads to abnormal glycogen with long chains and liver cirrhosis. A related past MCQ asks, glycogen stored is normal in all of the following conditions except, and the answer is Anderson's disease. Type phi, McArdle's disease, results from a deficiency of muscle phosphorylase. Patients experience muscle cramps and exercise intolerance. Past MCQ, McArdle's disease is due to the deficiency of, and the answer is muscle glycogen phosphorylase. Type 6, Her's disease, caused by liver phosphorylase deficiency, leading to mild fasting hypoglycemia. Type 7, Taruy's disease, due to phosphofructokinase deficiency, resulting in hemolysis and muscle weakness. Biochemically, the rate-limiting enzyme in glycogenolysis is glycogen phosphorylase, another common MCQ. Here are some additional frequently asked UHS MCQs. Which coenzyme is present in muscle phosphorylase? The answer is pyridoxal phosphate, or vitamin B6. Gluconeogenesis from glucose 1-phosphate requires. The answer is phosphoglucomutase. Which enzyme is common in both glycolysis and gluconeogenesis? Also, phosphoglucomutase. UDP glucose is converted to UDP glucuronic acid in the presence of. The correct answer is NAD+ and UDP glucose dehydrogenase. Thiamine pyrophosphate isn't directly involved in glycogen metabolism but is essential in other glucose-related pathways. So let's wrap up. Glycogenesis stores glucose efficiently using glycogen synthase and branching enzyme. Glycogenolysis mobilizes glucose quickly using glycogen phosphorylase and debranching enzyme. This entire process is regulated by hormones, allosteric signals, and enzyme phosphorylation.